I'll be talking a little bit about genetic summary data, substructure, and metabolomics, derived food biomarkers, among other things. So let's make sure we can click through. There we go. So hopefully all of you know this. I'll just skip through pretty quickly. And I have nothing to disclose. Okay, so let's start with just looking at some of the omics data that we have. So if we start with DNA, we have millions of genetic variants and markers, and we can gather this in what we call genomics. And if we have transcription, then we can look at mRNA, and we can gather transcriptomics. Then translation, we can get our proteins and look at proteomics. And then we can have enzymatic reactions and gather compounds, many of these endogenous to our body, but some of them coming in from outside, so exogenous compounds as well. And we can gather all of these in metabolomics. And all of these influence our phenotypes. And something that I think is really interesting to highlight is what we might consider the environmental influence through this omics pipeline, where it starts with little environmental influence up by DNA and genomics, and quite a bit of environmental influence as we go through. Now, there's a lot of omics I'm not even covering here, like methylation and so on. Today, I'm going to be focusing on genomics and metabolomics. So what does my team do? We do a balance of applied and method development research. So we get to work with a lot of wonderful collaborators to identify the genetic influence of health and disease, and to understand what I like to consider the biological why behind healthy diets. A lot of the work in our team in software and method development is to improve the utility and equity of genomic data. And more recently, we've been building pathways and pipelines to create a more representative group in our community. So today I'll focus on these two. So there were 3 learning objectives that you will all get, hopefully, from today to identify opportunities and challenges of using genetic summary data, to discuss reasons for underutilization of publicly available admixed genetic data and its role in propagating inequities in research and precision medicine, and to describe the utility of metabolomics-derived food biomarkers in research and the clinic. So first, talking about increasing the utility and equity of genomic data. So we've had amazing progress in genetics of health and disease. If we look across our genome, we can see a little lollipop here, and this is an association between a genetic variant and a trait or a condition. Now, this was in 2005, just at the beginning of the genome-wide association era, and in 5 years later, we had hundreds of signals, and 5 years later, thousands of signals, and even more now. And if I were to show you this in 2023, it's even larger. However, there's both opportunities and challenges. All of this data that I just presented is available as summary level data. And so we have genome-wide association study data available on the internet right now. We can go and download it, no firewall, no privacy protections. There through the GWAS catalog, through things like the Pan-Ancestry genetic analysis of the UK Biobank. And there's other public large genetic databases as well. Allele frequencies are available through places like Nomad and Alpha through dbSNP, and large sequencing programs such as TopMed and the Genome Sequencing Program. Through biobanks such as UK Biobank and the more recent All of Us, summary data compared to individual level data is often easier to access and use. And so we're able to do a lot of really cool things with summary level data. We're able to use them as weights for polygenic risk scores. We're able to use them in causal inference, such as Mendelian randomization. And we're able to use them in genetic correlation, again, just with summary level genetic data. We're also able to prioritize putative rare causal variants when we have people come in with rare diseases, and potentially use them as external common controls for case control association tests. But there's challenges as well. So when we have summary data, it can mask within or between sample substructure. And this can be due to ascertainment differences within our groups, potentially due to ancestry population structure, or even differences in technology and processing, differences in data generation. And when we mask within or between sample substructure, This can cause bias and confounding, false positives. It can also reduce our precision and reduce the power of our studies. This is especially hard when we're studying genetically admixed populations and makes a robust use of this data very difficult. So this brings me to our second learning objective. And here is a figure from the GWAS Diversity Monitor, where I've pulled data from 2022. And on the left, we can see participants by ancestry. And the peak here is European ancestry. So we can see the participants in genome-wide association studies from 2022, over 90% were of European ancestry. It's a little shocking still in this day and age that it's so skewed. Less than 10% combined were from the other ancestral groups. So we have an underrepresentation of data. We also have an underrepresentation not only by ancestry, but other factors such as race, ethnicity, environments, conditions. And I also like to highlight that the data we have that is representative is also underutilized. And this is in part because of that substructure that I mentioned earlier. It is harder to control for this substructure and to avoid confounding and bias. In this, the underrepresentation and the underutilization propagates inequities in research and medicine. So here's an exemplar, a very common exemplar to show some of this inequity. So on the x-axis, I have different ancestral groups, European all the way to African. And on the y-axis, we have prediction accuracy. And what we're looking at here is the prediction accuracy of a polygenic risk score that is anchored to a European ancestry group. So that's why European is at 100% here. That's what the risk score is built on. And so we can see that not only does the prediction accuracy decrease for the other ancestral groups, but some ancestry groups have a much, much larger decrease in accuracy. And what this means is that if we're thinking about bringing these risk prediction models, polygenic risk scores, into the clinic, we need to be thinking about the differences in prediction accuracy across different ancestral groups. Now, in addition to having inequity or differences in the clinical utility by these different groups, I also strongly believe that underrepresentation limits our full understanding of the genetics landscape. And we can clearly see this if we look back at this GWAS plot from the GWAS Diversity Monitor. So again, on the left, we have the participants by ancestry. Again, over 90% are in this pink European category. And then on the right, we have all of the associations, the new associations discovered in 2022. And what we see quite clearly is the pink really shrinks and the other colors really make up a much larger proportion of this plot, so that over a third, even though less than 10% of the participants were from other genetic ancestry groups, over a third of our discoveries are from these other ancestral groups. And so by not studying these other ancestral groups fully, we're limiting our understanding. Additionally, certain conditions may not be studied much or at all if they occur more in certain groups than others. So representation is important. Studies of conditions that are prevalent in underrepresented groups are important. It gives us a better understanding of the entire human health landscape across genetics, genomics, environmental, and so on for everyone. So how does my team work to address this problem? We develop methods and recommendations and software to increase the utility and equity of genetic summary data. We work to enable rigorous and reproducible research for all samples. And we work to make the methods and the software easy to use. So I'm going to give a few examples of this. I want to start with the use of external common controls. So the motivation here is that we want to identify rare genetic variation that's associated with case or condition or disease status. But sometimes we have sequencing data for cases and no controls. Imagine a study where you're studying somatic mutations in cancer and you've just gathered case samples, but now you want to look at germline mutations and you want to compare them to controls. Or perhaps we have a control set, but it's just not a large enough sample size, because really we need sample sizes of 10,000 or even 100,000 for adequate power. And so in these situations, we might consider incorporating external common controls. So here's a visualization that we put together when we were trying to think about this. So on the x-axis, we have condition prevalence, so maybe rare diseases or conditions, uncommon or common. And on the y-axis, we have genetic architecture, from Mendelian all the way up to polygenic. And so maybe we're in the space where we have a very rare condition that's driven by one or only a handful of genes. Well, in these situations, we might do very well with family studies or case groups, and we might be able to identify the genetic determinants. On the other side, if we have a very common disease or condition that's influenced by a large number of genes in the thousands, well, then we can probably garner enough resources to complete very large studies. In the thousands, hundreds of thousands, there's some million-person studies that have come out recently for traits and conditions that are so large. But when we're in the middle, and I'm not even talking incredibly rare, I'm talking about conditions that might occur in 1% or 2% of the population, it can be more difficult to garner enough resources to have these very large studies that we need to have enough power to identify the genetic architecture. And so in these studies, we believe that external common controls can be highly valuable, although I will say they're used in all of these situations. And so if we're thinking about using another data set that's been generated differently, we need to think about differences in the sequence generation. Maybe we're using whole genome, high depth, or whole genome low depth, or whole exome. And what we can see is that depending on the sequencing technology or on the processing pipeline, the genetic variants that we get, here represented by those little yellow dots, are going to be different. Now, really what I'm saying is if we gather our cases over here and we generate our genetic data, and then we separately gather our controls and generate our data, and we bring them together to analyze, we are going to have differences in alternate allele rates, and therefore we're probably going to have confounding and false positives or false negatives. I think a lot of time people forget that we can also be missing things from bias. So what we did here is we said, okay, let's think about a traditional case control burden test. And here what I'm showing you is under our null hypothesis of no association, we expect nothing to happen, which means we expect what's happening on the left in our cases to be the same as what is happening on the right in our controls. No association. G star is our gene of interest, and lambda is just our rate of rare alternate alleles. So we developed proxy external controls association test. And on our top here, we can see it's very similar to our traditional case control burden test, except for now we're weighting by some kind of proxy in the bottom. And some mathematical niceness here, it turns out that if we make a distributional assumption for the Poisson distribution, it's a really fast and efficient test. So then the question is, what is the proxy that we want to use here? Well, we would like our proxy to be able to capture differences in the rare alternate alleles in each gene region that might be due to differences as in sequencing technology and pipeline. Maybe we could use depth of coverage, or maybe we could use the number of rare or non-functional synonymous variants. And that's what we did. So here is a schematic to kind of highlight this a little bit more. So on the top we have our cases, and on the bottom we have our controls, and we have this for a variety of genes. The red stars are our rare functional variants that we expect to affect the protein, and our dark circles are our rare synonymous variants, the ones we don't expect to have a functional effect on our protein. And if we focus in on gene 2, well, we can see there's many more red stars functional variants in our cases compared to our controls, and also our rare synonymous variants are about the same in our cases and our controls, indicating we're probably calling the region pretty similarly between the two. We believe this might be a true association. But over here in gene 3, we also see an increase in the number of rare functional variants, our red stars, but we also see an increase in our rare synonymous variants in our cases versus our controls. We believe this might be due to a difference in sequencing technology and pipeline, and that this is likely driven by a confounded association. So we updated our ProxyCat formulation, and on the top we have our functional variants, and on the bottom we have our synonymous variants. And in our cases compared to our controls, when there's no association, we expect these to be equal, and when there is one, we might expect more functional variances in our cases compared to the synonymous. This controls for differential gene bias in cases and controls. So we applied this in a bunch of simulations. I'm going to show you the work that we did in the real data. So we had about 1,000 whole exome sequenced children with severe childhood obesity and about 30,000 external common controls from EXAC. And if we look at our black dots here, this is a QQ plot where the x-axis is what we expect and the y-axis is what we observe. And in a well-controlled test, we follow this line way down here. So any huge systematic deviation from this line shows a lot of bias going on. We have a problem in our test. Our traditional test is in black here, and we can see a huge deviation from the line both at the middle and at the end. But our new test, ProxyCat, follows the line much more closely. So then we wanted to continue with this development, and we thought, what could we do a little bit more? And we looked at the general formulation of ProxyCat. And so here in our rows are our cases and our controls, and then our columns are our functional variants or our not functional. Now, if you're looking at this and you're saying, Audrey, I know what that is, that's a 2x2 contingency table, you would be right, it absolutely is, with one just little difference. Our observational unit here, the unit by which we gather and analyze our data, is our alternate allele, not a person. So usually when you do contingency tables, you're dealing with people, we're dealing with variants. Now you might also be thinking, I know how to analyze data from a 2x2 contingency table. And if you're thinking chi-squared test and a Fisher's test, you would be right. And so we thought, gosh, I wonder how those would relate to ProxyCat. And so we did exactly that. We looked at how ProxyCat, Fisher's exact test, and chi-squared test of independence were related. And here's our negative log 10 p-value, our test statistic. And so we can see that ProxyCat is very similar, although not identical to these other tests. Now, you may or may not know that a chi-squared test of independence is very related to our logistic regression, equivalent in some situations. And so we thought, well, if ProxyCat is similar to a chi-squared test of independence, then we can take a chi-squared test of independence and put it into a logistic regression format. And if you're like me, you love regression because it lets you do all cool things. You can adjust for covariates, you can adjust for other structure. Putting this into a regression framework is great. So that's what we did. We put ProxyCat into a logistic regression framework. Again, a little different from what you would usually do. Our observational units are alternate alleles, not individuals. And this opens up a lot of possibilities, including multiple case or control samples and allele or variant level covariates. Now this is work still in progress, being led by a PhD student in my team, Jessica Murphy, and we've looked at the type 1 errors so far, and we can see that it is well controlled both for ProxyCat and log-prox. We're really excited about this method going forward. ProxyCat is already out. The regression framework is not yet. Okay, so logistic ProxyCat has the potential to include multiple samples. It does not require but can use internal controls. And what we're really excited about is it has the ability to include many different types of samples, case only, case control, meaning that we might have new avenues for meta-analyses. It works exceptionally well for very rare variants, and again, has the potential for including covariates and adjustment. So now I want to switch to another method that my team has been working on within this space to identify and leverage substructure in publicly available summary data. So here we have the Nomad aggregation database, which again, you can just Google and get to right now on your computer from the Broad Institute. It has about 140,000 sequenced subjects. And version 2.1 has more precise information, both some finer scale within continental ancestry information, as well as certain groups like a control group or non-neuro group. Over on the left, I'm showing how Nomad groups different individuals of people and presents their data. And some of these groups are pretty homogeneous ancestrally, like European or East Asian, and some of the groups, such as in gray, we expect there to be significant substructure or admixture. For instance, we expect there to be both European and African ancestry in African Americans, European, Native American, and African ancestry in American Latinx, and who knows in the other group. And incomplete ancestry information can result in incorrect analyses and conclusions and incorrect prioritization of putative causal variants. And this has been talked about a lot by others. And I want to highlight one particular publication that was left with what I would consider a really hard choice. So this publication was looking at severe hair loss in women of African descent. And studying severe hair loss in women of African ancestry is very important. Non-European ancestries and conditions are underrepresented, they are understudied. If we look in the paper, they use the African group from Nomad as the only control set for their data. Now, I argue this is a suboptimal choice. They either had the choice to not use the resource or to use the best but poorly matched external data set. And the sheer fact that they had to make this choice exacerbates inequities. If this had been a European sample, they simply wouldn't have had to make that choice. They would have had a well-matched group. Although I'll say substructure exists everywhere, right, at some level. And so our motivation is to estimate population substructure and allele frequency data so that we and others can robustly use this genetic data, for instance, admixed. And so here's a schematic of what we do with ΓÇÜomics. So we have a person or a sample, and we can generate the genetic ancestry of that person or sample. There's a lot of very well-established methods that do this for individual level data. There are not methods that do this for summary data except for ΓÇÜomics. And so now we have a public data set, Latinx, and given ΓÇÜomics, we can estimate the ancestry proportions of that data. But we can not only estimate the ancestry proportions, we can adjust those ancestry proportions, the allele frequencies of that data set, so that they match the ancestry proportions of our person or sample, and we can make the comparison across these groups more robust. And we do this with a mixture model, again, where our observational unit is the alternate allele frequency instead of individuals. We find this with using least squares. We find our ancestry proportions, and it's a really quick model and allows us to do some really cool things like block bootstrapping to identify confidence intervals. Okay, so again, we did a lot of really great simulations. I'm going to show the application to real data here. We applied this to Nomad version 2.1, and we estimated the ancestry proportions for all groups and all subgroups. This is in our publication. We used 1,000 genomes reference data, African, East Asian, European, South Asian, and Indigenous American. We did this for the exome data and the genome data. And again, we used block bootstrapping to estimate our 95% confidence intervals. So what did we find? Well, if we look in the African-American Nomad group, we see about 84% ancestry from the African and a little over 14% from European. If we look at American Latinx, we see a large proportion of ancestry from European and Indigenous American and a moderate proportion from African and East Asian. And if we look at other, we see a little bit of everything, although it's primarily European ancestry. Now, the other three groups are mostly matching their expected ancestry, East Asian, European. And in South Asian, we do see a moderate proportion of European ancestry, and this is as expected due to an ancient admixture event between South Asian and European. Okay, so we've estimated our ancestry proportions in publicly available summary data, and now we wanted to look at how well we do adjusting our allele frequencies. So here we looked at an adjustment from an African American sample to 100% African. And here we're looking at that compared to a 1,000 genome sample of African ancestry. So how how close are we? Here's our absolute difference, and here's our relative difference. And on the x-axis, we have different minor allele frequency groups. The unadjusted is in purple, and the adjusted is in gold or blue, where we're just varying the reference group that we're using. Now what we can see is regardless of whether we use the absolute or the relative difference, that our adjusted allele frequencies are a lot closer to 0. There's smaller difference than when we don't adjust. We also see that it doesn't really matter what the reference allele frequency data is that we use. There's not much difference there. Okay, so then we wanted to go back to one of our motivating examples, and we reanalyzed the data from this study of severe hair loss. And so we took the gene and the variants that they found in their cases, and we grabbed the number of alleles in our unadjusted nomad, and then we adjusted the allele count for 100% African ancestry. And we recreated the tests that they did in their paper. And what we can see and what's expected is that our p-value, our strength of our statistical test, differs by whether we adjust for ancestry or whether we don't. Now I will say that we do not know the actual ancestral proportion of the case sample. It wasn't provided and we didn't have the data to gather it, but I do believe that it is not 100% African, and it probably lies between our unadjusted and adjusted. So I think the true results lie somewhere in between. If we had their ancestry proportions, we could match them exactly. So this paper came out in 2021. I really love to highlight that 7 students on this paper started with me as undergraduates, including first author Ian Arriaga Mackenzie, who was the 2021 ASHG Cotterman Award winner. So we've continued our work with ΓÇÜomics. And so the first paper was able to estimate global ancestry and to adjust the allele frequencies. But since then, we've been able to adjust for more fine-scale ancestry because, like I said before, substructure is everywhere at some level. And we can also estimate other genetic substructure, like a genetic predisposition to a condition. We can estimate local ancestry across the genome, and we can use this to complete a selection scan and also to adjust our allele frequencies. So we hope to have another paper come out within the next year, and all of these and the methods are coming out in the next version of ΓÇÜomics on bioconductor. Okay, so a few examples of what these do. So we went from having 5 different continental level ancestral groups, and now we have 50 fine-scale ancestral groups that we can estimate. This is in part due to the wonderful data produced from Nomad version 3, where they produced harmonized data with the Human Genome Diversity Project and 1,000 Genomes. Now, I will highlight that there is still significant underrepresentation of these fine-scale ancestries within Africa, where, by the way, there is more diversity in Africa than outside of Africa. So this is a huge underrepresentation, Indigenous American and South Asian. So while this is better, it's still not complete. So then we can start doing some really fun things where we can take a Latinx group where we have our continental level ancestry, and we can start estimating the fine-scale ancestry proportions and mapping how similar those ancestry proportions and groups are to each other. We can also, with our local ancestry, do a selection scan across the genome. So the top is for Nomad African American, and the bottom is Nomad American Latinx. Now we see some signals we would expect. We see the HLA region come out, which we expect it to do for selection. We also see a bunch of regions that are related to immune response, which is also something that we might expect to see come out. So IGKV right here and the TRIM region are both related to immune. So lastly, we're really trying to get into can we detect the genetic predisposition to certain conditions? So we took prostate cancer because it's highly genetic, and we took variants for a polygenic risk score from 2018, and we overlaid them with our Colorado Biobank, and then we applied ΓÇÜomics. So I'm going to walk you through what we see here. So anything blue is our observed data, and anything orange is our estimated. So we first look at females where we observe no prostate cancer, right? They don't have a prostate, they can't get prostate cancer, but they still can have the genetics that could predispose them to it. And we wanted to see how stable are their estimates over age at less than 40, 40 to 60, and greater than 60. Now we've seen some variation, but not too bad. We then looked at prostate cancer cases, so men with prostate cancer. Now you'll notice we have no estimate down here for less than 40. There were only 10 individuals. We didn't have enough data. But if we start looking at 40 to 60 or greater than 60, our estimates are very close to 100%. And maybe this comes down a little bit because we might expect environment to be starting to play a role as people age. Now, what was most surprising was when we looked at all men, and we looked at 40 to 60 and greater than 60. Now you expect to see the observed increase where you have very few men that have prostate cancer and when they're less than 40, a little bit when they're 40 to 60, and a lot when you are greater than 60, the blue bar increase. But what we don't expect to happen is the estimated genetic predisposition to increase, because your genetic predisposition should not be changing by age. We believe this is a sampling artifact due to the Colorado Biobank, and that some of these men are coming into the Colorado Biobank because they have prostate cancer. And therefore, they come to the CU health system, and then they get enrolled in the biobank, and that is their point of contact. And if we look, actually, you can see a huge increase in the number of men coming in from less than 40 up through 60, again indicating that there's some reason we're recruiting more men to the system. A colleague of mine said, well, that's weird, Audrey, and he went back and he had a person in his team look at whether the polygenic risk score for prostate cancer was associated with age in our biobank. Again, it shouldn't be. The relationship between genetics and age for prostate cancer should not exist. It's not age at prostate cancer, it is just age, right, of everyone, controls and everyone. There's a strong link, again, suggesting that more people are entering our biobank as they get older because of prostate cancer. Now, you might look at this and think, uh-oh, that's a problem, but we look at it and think, well, maybe we can start capturing selection biases in summary data with this method. So we're really excited to move forward with that in the future. Okay, so now I'm going to completely shift, and I said I was going to talk about metabolomics, and I am. And so I'm going to get away from genomics completely, because I do get to wear another hat, and this is something that's really fun. When I've been doing genetics and genomics for a very, very long time, it's what I got my PhD in, what I trained in my postdoc. I never thought I'd be working in nutrition, and then I started working with some wonderful people, and voila, it's a huge portion of what I do now. So a lot of what we do is investigating the biological why behind healthy diets. And so a lot of this work is done by a really great and wonderful team. There's a lot of really great metabolomics experts, a lot of great nutrition experts, and then people in my team in statistics, really all coming together to make this work. So their motivation here is that there is ample evidence that certain diets, such as the Mediterranean diet, are healthy. But we don't really know why. Which food compounds, by what biological mechanism, or for everyone. And I'm going to today talk about this one. So it's difficult to track and quantify foods consumed. It's especially hard in observational studies, although it's also difficult to track compliance in intervention studies, even when you give them the food. There's a potential for food biomarkers to quantify food consumption and compliance in research, and also to identify food and medicine interactions. I always think, how great would it be if we could just determine what people ate by a pinprick, and then in the ER or something, when we couldn't talk to them, we can determine if there'd be any adverse events. So food biomarkers, we use untargeted metabolomics of foods and of human biospecimens. Here I'm going to be showing plasma, but we've done urine in the past, and before and after consuming foods. So we are using a study by Dr. Wayne Campbell at Purdue. It's a crossover controlled feeding study with 41 individuals, 28 women and 13 men. around 46 years, and obese or overweight. It's a secondary data analysis, and we are gathering plasma pre and post each of our interventions. We are also gathering a variety of cardiometabolic health indicators, such as total cholesterol, HDL, LDL, and blood pressure. So we take our Mediterranean diet, and we have our plasma from our individuals, and we complete untargeted metabolomics on this plasma. Additionally, we gather the 100 foods that people ate on this diet, and we complete untargeted metabolomics on those foods. We bring these two data sets together to capture food biomarkers. And we do this in two different ways, one through a food-driven approach and one through a health-driven approach. And so here are these two approaches side by side with various members of my team and Dr. Emily Hill, a postdoc in the Foodomics team who's running a lot of these, leading a lot of this work. So on the left, we have the food-driven biomarker approach, and on the right, the health-driven. And what I want to take home is that a lot of the same things are happening in these two different pipelines, just at different times. And so in the food-driven approach, we start with a compound extraction in food, and then we pull out food-specific compounds, look into human biospecimens and their association with health. For the health-driven, we switch this, and we start by extracting the compounds in our human biospecimens first, and then we look at health association, and only then do we go back and try to identify those compounds in foods. So starting with the food-driven biomarker approach, we gather compound extraction in 100 foods that they consume, and we identify food-specific compounds present in just that target food and not in the other 99, so only this dark blue right here. And then we look at whether they're present in human biospecimens. So if we look at walnut up here, we can see that there's 1,400 compounds that were seen in walnut and other foods. They're not food-specific. But there was a little over 300 that were food-specific. And of those, 23 were also seen in plasma. Salmon has a little over 1,000 food compounds that were also seen in other foods, and almost 400 that were food specific to salmon, and 143 of those that were seen in plasma. So we are taking these foods forward, and I'm going to focus in on salmon here. And so we took forward those food-specific compounds in salmon, and we looked at whether they changed in plasma after someone ate a med diet containing salmon. And so here I have volcano plots where I have the estimate on the x-axis, and I have the strength of signal on the y-axis. So a higher signal is a stronger association. And we can see compounds identified through a lipid fraction, so lipid soluble, and through an aqueous fraction, so soluble by water. And first, you can see there's a lot more compounds that were identified by lipid than by aqueous. And this, we believe, is in part due to it being plasma and not urine, and also potentially due to salmon, the food that we're estimating. Additionally, over here, we can see these estimates are the ones that increased after consuming a med diet and pass a false discovery rate of 0.05. These are our putative biomarkers. So we take those forward and we look at them a little bit more in depth. So here's a heat map where our first column is diet. All of these compounds increased significantly over diet, and we looked at their association with change in health indicator. And so I want to focus in on 3 compounds that both significantly changed over diet and had a significant change association with change in health indicator. And this first one is most interesting because it strongly increased over diet, and it has a strong association with what we would consider a beneficial change in health indicator, lower cholesterol. And so this particular compound is a cardiolipin precursor with a possible role in cardiovascular health through mitochondrial mechanism. So now I want to zoom in on the other health-driven biomarker pipeline. So here we first do compound extraction in human biospecimens. And I put this up here to show the quality control pipeline that we go through. It's quite in depth. So we start with a lot of compounds that are identified in plasma, almost 3,000 lipid fraction and over 4,000 identified through the aqueous fraction. We remove compounds with high missingness, and we complete a batch and order correction. And then we want to keep compounds that have a low variability across our quality control samples. These are quality control samples that should stay consistent. We also only keep compounds that have a high variability across our individuals. If these compounds are stable across all individuals and across all diet intervention pre and post, I don't really care about them. We don't want to study them, so we're going to remove those. We code our NAs, our missing values, as zeros because zeros are important when we're studying food and we expect compounds to be there or not. And then we rank our data. So after this, we had nearly 2,000 lipid compounds and about 1,800 aqueous. We first looked at compounds that significantly changed at a false discovery rate over diet, and we also looked at compounds that are associated with a change in health indicator. We brought those together and we identified 485 compounds that both changed over diet and were associated with a change in health indicator that were lipid, and 7 from the aqueous. Now we wanted to focus on a little bit more and say, you know what, we're really interested in are compounds that increase over diet that are associated with med. So we looked at these a little bit more depth, and of these we had 108 that we would consider had a beneficial relationship or change with health indicator. And what I mean by that is this group up here both increased over diet and were associated with a decrease in total cholesterol. That's this group up here. This group down here also had an increase over diet, but were driven with a beneficial change by a decrease in triglycerides. We also had 44 compounds that increased over diet, but had what we would consider a deleterious change with a health indicator. So this group increased over diet and were associated with an increase in triglycerides, and this group down here was driven by an increase in cholesterol. So we focused in on the 108 compounds that increased over diet and had a beneficial association with a change in a health indicator. And then we annotated those compounds and looked at whether we could find them in the 100 Mediterranean foods. Now, before I show you those, I just want to mention we also looked at compounds that decreased over diet. Maybe these are ones that are seen more commonly in a Western diet than a Mediterranean diet, and were associated with a deleterious change in health indicator, and we're following those up later. So looking at these 19 compounds, again, they significantly increase over diet and are associated with a positive change in a health indicator. And just to highlight a couple, we see that this compound is seen in blueberries, chickpeas, kiwi, and so on. This compound here is seen in all those foods, as well as pork tenderloin. And so this health-driven approach is able to get compounds that are seen in a much larger set of foods, but might not be precise to individual foods, where the other food-driven biomarker approach is more likely to identify more precise food compounds. So we're following up the food-driven biomarkers with an intervention study currently in progress to assess the replicability of some of the putative biomarkers using untargeted and targeted metabolomics. So you can see a variety of foods that we've taken forward, and we're going to be looking to see whether we can identify those food compounds again. So hopefully by this point, you have seen that there's an abundance of omics data that provides a lot of opportunities that we can improve the summary genomic data use through having methods to have appropriate case control analysis using external common controls and estimating and adjusting for substructure, and as well as we can use metabolomics to identify biomarkers of foods and health. And I will end there and say thank you to my current team, collaborators, and funding agencies. And I believe I'm supposed to end with this slide. I'm happy to answer any questions. Yes.
So I was curious about one thing you said earlier in the talk about a cases-only approach to understanding sort of genetic substructure, and I could see this being relevant in pharmacogenomics, for example. How do you apply your methods to cases only, and there is it sort of internal control for non-synonymous, or I guess synonymous changes? I'm curious.
Yeah, that's a really good question. So the method that I presented would use external controls as a control set if you had a case-only data set. There are methods out there that are developed for case-only. They tend to work sporadically, but are most suited to Mendelian type genetics. And so I wouldn't necessarily recommend them with other ones. Although, unless you're studying cancer or something where somebody can be their own control. It's a good question.
Yeah, great presentation. I have two questions. First, about substructure again. I'm going to get the details wrong, but I remember there was a GWAS paper in Finland where the gist of it, they showed the entire association could be mapped to specific regions of sort of microethnicities.
Yeah.
So is that something that we can control for at this point, or is it just sort of these macro, South Asian, African, et cetera, or are we to the level where we can control for that? That's my first question.
Yeah, it's a great question. So ΓÇÜomics version 2, which will be coming out hopefully soon, does start to control for more fine scale ancestry. Now we have to have that fine scale representation to be able to control for it. I will just mention that if we have complete stratification, then we can get rid of our signal completely if we adjust for it. So within that situation, we might want to be looking within instead. And so that's something that I think the whole genetics community is a little, if you have complete stratification by ancestry and phenotype, well, then there's no separating them. And I'm not really sure what to do. Yeah.
Good. And my second question is related to your prostate cancer polygenic risk score work. It's of personal interest. We're actually developing a polygenic risk score test right now, including the prostate cancer PRS. There's a very talented faculty member, Virtue Darst, who's leading on that. So my question to you is, us as laboratory medicine physicians and genetics, what should we be watching out for? We're very reluctant to get into this business because of all the sort of issues you've highlighted of how you can get it wrong. And we've always been inherently skeptical of using any GWAS associated variants for any clinical test because of all the problems you've highlighted. This is the first time we're getting our feedback. What should we be doing?
Oh, sure. So my recommendations, if people are developing polygenic risk scores for the clinic, and so there's two things that I like to keep in mind. Anytime we're developing a risk score, polygenic risk score, any kind of risk score, machine learning, we should always be assessing the prediction accuracy across a variety of groups. So prostate cancer, you can't really assess across different biological sexes, but in general, you should evaluate by sex, by race, by ethnicity, by socioeconomic status. We should make sure that we don't have different prediction accuracy based off of those groups, or if we do, we'd better be upfront about it and be very clear. Additionally, I always like to think, what is the clinical utility for the risk score that we're developing? How is it going to change the clinical care of practice? I was talking with somebody earlier today where I think, people developing a clinical utility for a diabetes risk score seems to me to be really interesting, because there's those few people in the middle where they have indicators for both type 1 and type 2 diabetes. The treatments are very different. And how do you know if somebody comes in, which one they are? Well, in that space, a polygenic risk score could maybe push you in one direction or another. And so that's also something I would be asking, is how is this going to change the clinical care? Yeah.
So, wonderful work. What type of power hit do you take when you're doing this? That stratification, when you're doing that correction.
For which, the correction for which context?
For when you're using the controls and then adjusting for
Oh, for the synonymous variants, the common controls. It's a really good point. So if you have the best study design is to gather your cases and controls together and to sequence them together and generate your data together, you are going to have more power and less confounding, absolutely. So compared to that, we are definitely less powerful, right? There is no consideration. Compared to not being able to do the study at all because you don't have controls, we have a lot more power than that. And then also being able to incorporate more data robustly. We also have, we can get over the power hit. So it's a really good question. There's also another wonderful method that I didn't have time to talk about from some collaborators called ICAT, where if you are incorporating internal controls alongside external controls, that's a really great way to do it. Mm-hmm. Sure. I think there's 3 in the chat. Or Q&A. Oh, there's, 3 in the Q&A. Okay, so I have 2 questions. I don't think we're unmuting. Oh, same person. Okay, first question. Doesn't the first method of weighting based on non-synonymous substitutions assume no genetic drift is taking place? But if drift is likely in the smaller group you look at. So the assumption of ProxyCat where we are having functional weighting by synonymous makes the assumption that 1, the signal is in functional, not synonymous. Whatever we're using as our proxy is not where the signal is. If the signal is there, we have a problem. Additionally, it's making the assumption that any systematic differences we see between cases and controls is not going to be different between our functional and synonymous, which I think actually is a pretty good assumption. Overarching, we don't expect sequencing technology or computational pipelines to preferentially call functional versus synonymous variants. But absolutely, if we have a signal that's in what our proxy is, this test is not going to work very well. And the second question was patient plasma-driven metabolomics approach. A metabolomic marker may change. It may even be associated with health, but how do you know that diet caused the change and not the patient? So that's an absolutely great question. Something I did not have time to show is that when we're studying metabolomics, we have compounds that are metabolized, and then we have metabolites, right? And so the whole metabolism of metabolomics and compounds really makes it complex to study. Now, one of the reasons why we focused in on compounds we see in the food and also in plasma is because those are the compounds that are less likely to change. Now, I said less likely, I didn't say didn't change, because there's definitely, we saw, because we looked at the metabolites, the predicted metabolites, there were some food-specific compounds that were also predicted metabolites from some of the other food-specific compounds. So it's really complex. But they're more likely to be there because they didn't change. And if you're trying to develop a food biomarker, well, then it can be really helpful to have a compound you see in the food that travels through to the urine or the plasma. So yes, very complex, and it's one of the reasons why we're doing the follow-up study to see if we see them again. Great. Yeah, thank you.